In this video, we're continuing a discussion of meiosis. In the first video, we talked about how we send a diploid cell at the beginning of the process through this meiosis process and make haploid cells at the end. In this video, we're going to try and extend that conversation by really talking about how you're constantly doing this. So males and females are constantly making new sperm or new egg, but every time you go through this process, you get cells at the end that are sort of a different genetic half of yourself every time, and that's because there's a step in there that allows for a lot of mixing up. And so we're going to see that there are really two mixing processes. One's called independent assortment, and one's called crossover, and we'll take them one at a time. So independent assortment is this idea that when you line the homologous pairs up side by side, that's really the key step in meiosis 1 where both mixing events occur. The idea is that because you bring together the two pairs side by side, you can line them up two different ways every time you put them together. And so to help you see this, I'm going to go through kind of several different lineups here. I've got my organism with 4 homologous pairs. Unlike the first video, I also tried to show one of the pairs being a little darker, and let's say that that's the chromosome that this organism got from its father. Remember that we all have homologous pairs because we got one set from dad, one set from mom. And so the idea is, okay, now that this organism is making its own sex cells, will it pass on its dad's chromosome for that set or its mom's chromosome for that set? And the idea is that, again, every pair is independent of the other pairs. So remember that after we line up the homologous pairs, we split them in meiosis 1. So I'm just using that blue line to indicate separation. In meiosis 2, the X copies get split up like this. So I'm kind of hoping you can imagine that each column here is like a separate gamete then. And that these two gametes would have this combination of alleles, and that these two gametes would have this combination of alleles. So that's kind of maybe one way we could potentially pass on the chromosomes. You'll notice that I just happened to line up sort of all of the chromosomes that we got from mom on this side and all the chromosomes that we got from dad on this side. So, one possibility is that you just pass on all of your dad's chromosomes to a set of gametes, and maybe you pass all of your mom's chromosomes onto a set of gametes. But if you were to go through this process again, then the next diploid cell that goes through this process might line them up a different way. You'll see that I kind of kept most of the pairs the same, but that I flipped these guys around compared to the first example. So if I split the homologous pairs in meiosis 1 and I split the exact copies in meiosis 2, you'll now see that these two gametes have this combination of letters now. Notice that that's different from what I had over here, and that these two gametes will have this set of letters. And that's a little different from what I had over here. If I were to keep doing this, you could see that maybe there's yet another way I could line this up. If another cell starts the process of meiosis, maybe I just happen to line up the homologous pairs this way in meiosis 1. You'll notice that compared to the second example, I just flipped these guys around a little bit. So if I split them up, meiosis 1, split again, meiosis 2, now I've got this set of alleles in these two gametes, and these two gametes will have this set of alleles. And really, what I want you to see is that all 8 of these are a little different from each other. They're passing on a slightly different half of themselves each time. And at least in these two examples, the idea is also that you're kind of passing on a little bit of what your mom gave you and a little bit of what your dad gave you for different chromosome pairs. So just in case this is a little bit kind of hard to understand, just a metaphor that might help you from the real world. The one example I can think of is sort of like a tic-tac-toe game commonly found on playgrounds, where you can kind of spin around the block and get an X or get an O. And so the idea is that each spinner can be spun either of two ways. You can either get an X or an O, and each little spinner is independent of the others. And so what we're saying is independent in our chromosomes here is that the chromosome pairs are independent of the others. These guys don't like watch what these guys do and then imitate them. They can line up independently of the pairs above or below them. And so here's kind of my way to try to explain that with words, in case you want to pause the video and jot this down. The key is that because we're lining up the homologous pairs side by side, we can line them up two different ways every time we line them up. And so every cell that starts this process might line them up a different way and get different end results every time. Okay, so that's independent assortment. And the second mixing up process occurs at the same time. It's also due to the fact that we're lining up the homologous pairs side by side in meiosis 1. And the idea is that while they're side by side, they might get so close together that the inner tips might actually cross with each other and kind of switch places at their tips. And so it's really hard for me to show this with kind of animation, but I'm just going to show this kind of migration here. And this idea that maybe the little bit of red, maybe from what Dad initially passed to this organism, is now mostly on this chromosome with a lot of what Mom passed on, and kind of vice versa. Now there's a little bit of what Mom gave us on this whole chromosome here, which most of the rest of it is what Dad passed to us at one point. And so the idea is if we once again go through meiosis 1 and 2, the idea is that all four of these gametes are now a little bit different from each other genetically. So what's cool about crossover is it mixes things up even more, and as it turns out, sort of how much the tip switch could be different each time as well. Here's an example where a little bit more crossover occurred. Sometimes no crossover at all will occur. So how much kind of mixing up occurs from pair to pair might differ as well. And so again, maybe just the broad idea of crossing over is that it's the inner tips of homologous pairs, while those X's get so close to each other in that side-by-side lineup that maybe the inner tips can kind of switch a little bit and mix up what you send ultimately to those different gametes. All right. So, I just want to finish this video by talking about how both of these things that we just talked about are only possible during meiosis because of the side-by-side lining up of the homologous pairs. None of this is possible in mitosis because these homologous pairs are never side-by-side, and so you can't, what's the only way you can line this up with in one straight line? You can only line it up one way. And that's because in mitosis we're trying to make exact copies, so this works great for its goal, which is to make an exact copy of the initial cell, but this process works great for generating genetic diversity. All right, so in this video we just kind of talked about the two mechanisms that enable genetic diversity during meiosis. And remember that meiosis is the step leading to sexual reproduction, so this is also how we get diversity in sexual reproduction overall.